my name is Emily Linkevich, and I'm an undergraduate student at Albright College. This summer, I worked on a research project studying the effects of the non-native Microstegium vimineum on small mammals and insects. As stated before, this study focused on the effects of Microstegium vimineum on small mammals and insects. However, we primarily looked at one small mammal, the Peromyscus leucopus, more commonly known as the white-footed deer mouse, which you can see what it looks like in the upper right-hand corner. These mice are habitat generalists, meaning they have a wide range of habitats in which they can live and a variety of foods they can eat. While the opposite, a habitat specialist, has a limited habitat range and limited diet. As generalists, these mice have a varied diet, but Wolf and others most commonly identified arthropods in the stomachs of these mice, which added to my interest in the insect diversity of the research site. And I'd like to give you some background information on Microstegium vimineum, more commonly known as Japanese stiltgrass or Japanese packing grass. It is native to Japan, China, India, and other Central Asian countries. It was first documented in the United States in 1919 in Tennessee, which it was most likely introduced by accident as it was most commonly used as packing material for porcelain. Since then, it has slowly made its way up the eastern coast of the United States. It has been observed in Nolde Forest just outside of Reading in the trapping grids and sites by Dr. Osgood and others. I'd like to give you some helpful identifiers for this plant. It typically grows from 60.96 to 106.68 centimeters. It somewhat looks like thin, wiry bamboo, especially if you look at it from the stem. A key identifier for this plant is a silvery stripe on the midrib of the leaves, which somewhat glistens in the light. You can see what the midrib looks like in the picture to the right. The leaves range in length from 2.54 to 7.62 centimeters. It has parallel veins rather than webbed veins, and it's a shade-tolerant species, which means it can survive in low light levels, which helps it thrive in closed canopy conditions. I'd like to go over some of the previous literature that I looked at for this project. Medicoff and Emory found that insect populations tend to increase in the presence of Japanese stiltgrass, while the native plants decrease. I also found that large mammals will avoid areas in which this grass is present until it's more comfortable with the plant, which led me to wonder how Peromyscus leucopus and other small mammals would react to the presence of Japanese stiltgrass. I also found that select cut and clear cut logging influenced mice populations, along with this causing the stiltgrass to become a dominant plant over the native populations. I'd like to do a basic overview of the methods that we used. The site that we chose was Nolde Forest Park, just outside of Reading, in which we used two different locations in the park to capture mammals and insects. The clear-cut control for the mammals had 25 traps in a 5x5 grid, while the select cut had 64 traps in an 8x8 grid. These traps were opened twice a week and checked twice a day to limit the stress on the animals. They were baited with sterilized seeds and peanut butter rolled oats. Data recorded for the mice included ID tag number, sex, age, reproductive status, and parasites. While the insect data, we set up fly paper and pitfall traps 3 times within the time period we had at random locations within the grid. At the select cut site, we had 10 traps, while at the control site, we had 6. We then left the traps out for a week, and then after a week, we retrieved them and brought them back to the lab. Then at the lab, with the help of a microscope and dichotomous key, we identified the insects by order. For the data analysis of Peromyscus leucopus, we started with 728 data points. We then excluded the recaptures, which gave us 489 unique individuals. The primary animal found in the trap was the white-footed deer mouse. However, we found other animals, which included chipmunks and shrews. Continuing on with the data analysis for Peromyscus leucopus, we then put it into our studios, which we looked at the estimated population size. First, the select cut site had approximately 152 mice, while the control had approximately 47. We then looked at the estimated births for each period in those sites. The control for the entire study period had relatively small numbers of births and were relatively similar to one another, with the highest being 9.45 standard deviation of 3.79, while the lowest had 2.19 standard deviation 2.23. While the select cut, on the other hand, fluctuated quite a bit, where the births decreased from period 1 to period 5 and then increased from period 5 to period 7. Lastly, for the Peromyscus leucopus data analysis, we looked at survivorship and population. For survivorship, the select cut, like the birth rates, fluctuated greatly between all 7 periods, while in the control site, it mainly stayed within the 70% range for survivorship. However, between period 5 and 6, it decreased to the 60th percent range, and then it increased again for period 7. Population-wise, the select cut from periods 1 to 3 increased, then decreased for the next 2 periods, then increased again until period 7. The control, however, went in a pattern of increase, decrease, then increase again for the 7 periods. For the data analysis of insects, we originally had 10 traps on the select cut site. However, we could only use 9 out of the 10, as one of the pitfall traps was destroyed during the week it was left out. Even with losing one of the traps, we still had enough data to continue the analysis. Out of the 1,285 insects found and identified, the most commonly identified one was that of the acari, which are mites, ticks, and spiders, and diptera, which are mosquitoes and flies. Although the bar plot shows that unknown insects were the most prolific in our data set, the unknowns were just the arthropods that I could not identify with a dichotomous key, as they were in pieces or too heavily decomposed to identify with one, even with a microscope. Our RStudio analysis found there is no significant difference between the insect diversity in the control and the select cut. Now, this is with the unknowns included in our data set, as even when we took the unknowns out, the p-values were still relatively similar. The results of our study found that small mammals were more likely to be impacted by Microstegium vimineum than that of the insect diversity, which is contrary to what Medikoff and Emery found, which found that Microstegium vimineum increased the diversity in insects within a set population. Although our data did not find a significant difference between the select cut site and the control site of whether or not insect diversity was affected by Microstegium vimineum, this could be due to the fact that we had only collected insect data 3 times throughout the summer, giving us a limited amount of data on the insects collected. Although I've completed most of the data analysis and techniques, I'd like to do the seeker technique, which will help me look at the mice's movement throughout the trapping grid. However, due to current time limitations for the project, I have not completed this part. I hope to have this finished at some point before October, in which this project will be presented in front of a conference, and I'm hoping to do another acre next year to see if the Microstegium vimineum is truly affecting insect diversity and the mice, and do a more in-depth analysis. This study was funded by the Hearst Foundation, and this project was accepted by the Albright Creative Research Experience, otherwise known as ACRE. We'd like to thank Nolde Forest Environmental Education Center and the Pennsylvania Department of Conservation and Natural Resources for the access to the field site, as well as Ryan Brett for his help in the field research, as well as Dr. Mech, who helped me learn new techniques on animal trapping and new data analysis techniques.